Hi everybody, welcome to the last video in the innate immunity lectures. In this video, we're going to talk about inflammation, which is really the hallmark of the innate immune system. This is where a number of the cells that we've talked about previously are all going to come together and try to stop invading pathogens from getting into the body and growing to high levels. Let's take a look at how inflammation works. We're mainly going to be focusing on acute inflammation in this video. This is that key process of the innate defenses, and its whole purpose is to do a couple things. The first is it mobilizes those phagocytic cells and it gets them to the site of an injury. So it says, here's where the injury is, here's where the invading pathogens are. We have to get all those immune cells there to that area. The second is we contain the pathogens in that area and we eliminate them. So the phagocytic cells are able to destroy the pathogens themselves. And then the third part is to repair the tissue damage, to fix any of the problems or the damage that had happened during that injury. It's that last part we're not going to talk about in this class. So the last part is a little bit more physiology based. We're really going to function more on the elimination of the pathogen itself. Okay. There's typically what we consider 4 cardinal signs of inflammation, although your book lists them as 5. The 4 main ones: redness, heat, swelling, and pain. And then the 5th that's associated in your textbook is altered function as the tissue gets damaged. Okay. Keep in mind that inflammation happens in response to tissue industry, pathogen attack, but also trauma. If you sprain your ankle, if there's chemical damage, if you burn yourself, all of those things will damage tissue and cause inflammation. But for this class, we're really focused on the pathogen attack and what's happening in those types of inflammatory processes. Okay, so we're going to walk through the process of inflammation. So here we have a blood vessel. And out here is the tissue. So remember which cells reside where. So in the blood vessel, we have our neutrophils. We also have basophils and eosinophils, but remember the neutrophils are the real workhorse. There's really the only ones we're going to talk about in inflammation. And then out here in the tissue, we have macrophages. Okay, that's the abbreviation for macrophage, just so everybody recognizes. We have dendritic cells. And we have mast cells. And here's the edge of our tissue. Okay, so these are the 4 main cells that are going to be involved as we're looking at this picture. The dendritic cells we're just going to talk about tangentially because they're really going to connect what's happening here in inflammation to the adaptive immune response, which we'll talk about in the next unit. So something that breaks the skin, now you step on a nail, you get a cut, that's going to introduce bacteria into the tissue. So 2 things are going to happen, and they can happen in either order. So this doesn't have to happen with one cell responding first. It's really just whichever one was closer when the bacteria or the pathogens invaded. So what's going to happen is as these pathogens are coming in, the macrophages are going to do phagocytosis. And in that process, remember, they're going to be destroying the cells, but they're also going to be releasing cytokines. The mast cells will also be interacting with those bacterial cells. And remember, mast cells don't do phagocytosis, but they do have the PRRs that interact with the PAMPs. And so when they bind to the bacteria, that causes them to release their granules. And remember, the key thing here is histamine and what we call vasoactive factors. So these are various chemicals that are going to affect the vascular system. And the 2 big things that the mast cell is going to induce is vasodilation. So the blood vessel is going to dilate, it's going to get bigger. That's going to increase blood flow to the area, which means we're going to bring in more neutrophils as blood flow is now increasing through this blood vessel. And then we're also, in part of that vasodilation, going to see an increase in vascular permeability. What that means is that along here, remember, you have your endothelial cells that have been lining the blood vessel wall. Well, when we see an increase in vascular permeability, we're going to start to see spaces happening in between these cells. These spaces are going to be important because what they're going to allow for is fluid to leak out. And remember, what's in that fluid are things like complement proteins, which will now enter. So as the fluid has leaked out, complement has entered, which can come over and start helping to destroy the invading bacteria. And the neutrophils themselves can now squish out between these spaces. So they actually fold themselves out and they're going to move into the tissue. That process is called extravasation, and it's got a number of steps to it, but this key part right here is diapedesis. That's when the neutrophil actually squishes itself out in between the lining of the blood vessel and enters the tissue. This is when the cytokines are going to come into play. Remember those cytokines? These particular ones are chemokines, which means they're chemotractant. And so we've got all these cytokines the macrophage has been producing, and as they diffuse away, they get lower in concentration. The neutrophils that have left are going to follow that path until they find where the site of infection are. So now they're going to join in the fight with the macrophages. We'll have our neutrophils here. They can also do phagocytosis, or they could release their granules and release all those bacterial killing molecules. So these are the main steps of inflammation. And remember, we've got vasoactive factors in histamine being produced from mast cells that lead to vasodilation and an increase in vascular permeability. That gets cells to the site of infection and gets them out of the bloodstream. And then they follow that path of cytokines that was produced by the macrophages so that all those immune cells get to the area of the infection and start battling that microbe. These increase in vasodilation and vascular permeability is also where we get those cardinal signs of inflammation. So the increase in vasodilation is going to cause a redness because of the increase in blood flow, and heat because the more blood flow, the more friction, and so that tissue warms up. Make sure to note that this is heat of the tissue. This is not a fever where it's affecting the whole body. If you ever have swelling of inflammation where you get a little red, if you touch that tissue, it feels a little bit warmer. That's the heat we're talking about. And then the increase in vascular permeability, when this fluid leaves the blood vessels, that's what's causing the swelling. And you're also going to get pain affected here as that fluid is pushing on nerve cells. And there's also signaling molecules that are going to be produced through this whole process that help signal nerve cells, just to associate that with something going wrong in the tissue. Now that we've done an overview of all of inflammation, I want to zoom into the blood vessels and talk a little bit more about that process of extravasation. Okay, this is when the neutrophils, remember, are going to squish out of the blood vessels. So vasodilation has happened. We're going to get some spaces here in our blood vessel walls, and those cells are going to squish out from those spaces. Here's our neutrophil. And remember, the blood's been flowing, so these neutrophils are moving through the blood vessel. In order to do extravasation, they're going to need to slow down first. And so the first thing that happens here is that neutrophils are able to bind to molecules along the endothelial cells called selectins. So here's this molecule called a selectin, and the neutrophil is going to bind to it. And there's another selectin here, and as the cell moves along, it's going to bind to that one too. And so this binding is causing them to slow down, so they're not traveling quite as fast. They then form even tighter binding through adhesion molecules that are almost going to completely stop the cell. So we have another set of molecules here. They can bind to the neutrophils themselves. Those neutrophils have molecules called integrins that bind to these adhesion molecules and really stop them in their tracks. And now that they've stopped, they're now able to squeeze through and complete that diapedesis process. And the actual squishing through the endothelial wall. Remember, once they're now on this side, they're going to go and follow that gradient of chemokines or cytokines to wherever the site of infection was, to wherever our bacteria were. This process is extremely important because the macrophages out in the tissue can't destroy all of the bacteria on their own. They really need the neutrophils to come in. They're the main killing mechanisms that are really going to fight off those pathogens in the innate system. Now remember, we said acute inflammation had 3 parts, and that last part was really going to be resolving the damage. That's the part we're not talking about in this class, but it's still a really important part of the inflammatory process because the neutrophils, as they're releasing those granzymes, those enzymes that are going to be damaging molecules, they also damage your cells. So things like perforin will bind to bacteria and destroy their cell membranes, but they can also harm our own cell membranes. So we're damaging some of our tissue through this acute inflammation process. What that means is we have to have a way to shut those neutrophils off and then fix the tissue damage once the pathogen is gone. If that doesn't happen, we end up with a condition that's called chronic inflammation. This is when the inflammatory process keeps going, neutrophils keep coming into the area, macrophages continue to be there, and it's usually due to the persistent presence of some sort of foreign body or the cell thinking that there's still a pathogen there. When we see this, we see a lot of macrophages come in, and we actually will activate the adaptive system, and some of your lymphocytes, your T cells, will come into the area too. But they're not really doing anything for a pathogen. They're really just damaging host tissue. And we actually can see that when we look at the tissue themselves. So this is a granuloma. These are big lumps of cells that have walled off inside this normal tissue. So this is all normal tissue. But this is now non-functioning in the granuloma. So you get these big masses that get in the way of how the tissue can function. We tend to see these in things like hepatitis, and if the granulomas get really big and are a concern, sometimes they're going to require surgical excision to remove them.